So, good afternoon, everybody. My name is Pranati, and I will be teaching you about immunology and animal biotechnology, which is in your sixth semester, basically. So, without wasting further time, what I'll do is I'll just start with the classes, and then as you know, there are around 12 topics, and we've got only 8 classes. So, more or less, I will be covering two topics in a class, and then hopefully by within 8 classes all the 12 topics will be finished. So when it comes to the basic concepts of immune system, am I audible?
Yes, ma'am.
All right, sure.
Yes, madam.
Cool. So yes, please mute yourselves. And when it comes to the basic concepts of immune system, you get to see that we have to have an ability to resist whatever kind of infection or whatever kind of a pathogen that comes against us. And whatever we breathe a lot of smoke pollution. At the same time, we breathe a lot of viruses, bacteria for that matter, pollen grains. So all this goes inside us. So how are we able to stay as we are without getting infected or affected by them? That is where your immune system comes into play. So today the objectives that we are going to see is what are the components of immune system and what are the types of immune system. We do not have only one type of immune system. We have three types of immune system basically. And then we are going to see how blood cells are formed, and then what are the cells of the system that are involved that give us the immunity, and then we are also going to see the organs that are involved in giving us this immunity. Immunity is something wherein we are getting it by birth as well as we are acquiring it. So innate and acquired. We'll come to that in a minute. Let me share my PowerPoint presentation with you. Just give me a minute, please. Right. So when it comes to an immune system, the first thing is you should ask what is immune system. So it's a biological structures that are processes and within an organism, that is within inside us, that help us protect against a disease by identifying and then killing the pathogens and the tumor cells also. They also happen to be they might be present within the cell, within the body, but then they are foreign in nature. There's something against, or they're different from your normal cells that are present. So immune system detects a wide variety of agents from the viruses to parasitic worms and needs to distinguish them from the organism's own healthy cells and tissues so that they'll be able to perform properly. So immunus means exempt, refers to the foreign agents. And when it comes to the definition, the integrated body system of the organs, tissues, cell products, so that you are able to distinguish between non-self and the self, and whatever is non-self should be able to be neutralized you from like if at all they're pathogenic organisms or whatever for that matter, any foreign body, they have to be neutralized. So this is the dictionary from American Heritage. Stedman's Medical Dictionary has given this particular definition for immune system. So classification of immunity, you get to see immunity again is of three types: innate, acquired, and then you have passive and active. We'll come to that later. So when you see innate immunity is of two types. Sorry, immunity is of two types: the innate immunity and the adaptive immunity. Again, when it comes to the adaptive immunity, it can be natural and artificial. In your book, please don't get confused. There's nothing wrong in it. In your book, there are given three types of immunity. The first is the two types of immunity are innate and acquired. And then in the acquired itself, and then you also have the, active and passive immunity. We'll come to that later. It's a type of immunity. So innate immunity is where it is not exactly specific. So you're born with it. Within. Innate means from within. So it includes, the physical barriers, physiological barriers, cellular barriers, as well as cytokine barriers. Adaptive immunity is again natural, or in this again you have passive. So that is you're getting it from your mother, that is the maternal. Active infection means it is from the infection. You're exposed to the flu virus. So the immunity that is developed because of this is known as active immunity. And again you have artificial. Artificial is because of antibody transfer. That is the kind of immunity you're acquiring because of the transfer of antibodies. And then active is immunization. All your vaccines are artificial immunity. You're either giving a live pathogen or inactive pathogen, or a dead, be it a virus, be it a bacteria, be it anything for that matter. You're giving it in an inactivated form wherein it does not cause the disease, but your cells within the body will be able to recognize them and distinguish and tell that they are foreign in nature, so we have to act against them. So that comes under artificial active immunity. That is your immunization, or all the vaccines come under this category. Right? So when it comes to that, again, immune system has a lot of cells that are there inside our body. They can be your T cells, B cells, all of them, your basic barriers. So before that, we will come to the, see, again, what are the kind of threats that we have? There are viruses, carcinogens, that is cancer-causing agents, the pollution, the germs, then you name it, the toxins, fungi, bacteria, then parasites, your tapeworm, all of them for that matter, your nematodes, cestodes, all of them also happen to be parasites, your malarial parasite. So the barriers that we have within our body is basically you get to see that these are the invaders, these are the defensive mechanism that is present inside our body. The tonsils, the lymph nodes, the thymus, all of these. So again, the same kind of definition. Immunity, you have innate, non-specific, immediate. So this is immediate in nature. Whatever innate immunity you have, it is going to act against any kind of pathogen immediately. But if it is an acquired immunity, you first have to see that the antibody or the pathogen has to be recognized. Then it is going to be dealt with. How is it going to deal with that particular foreign body or a pathogen or a virus for that matter? It can either be a humoral immune response or a cellular immune response. Humoral is all your antibodies that are produced against that particular antigen. We will come to these later again. And when it comes to the cellular immune response, you have the T cells, your B cells, and for that matter, your natural killer cells, your null cells, all of them come under the cellular immune response, your new eosinophils. We'll come to that later. So this is all about the evolution of the immunity. So again, function is self and non-self discrimination. modified altered cells. And when it comes to the infection and immunity balance, you get to see that if you have a disease, so that is infection into virulence by immunity, one of the definitions. So what are the effects of the immune system? How is it beneficial for us? You get to see that we have protection from the invaders, and also you have this altered self elimination. That is, if at all there are any kind of tumor cells that are being formed in the basic, in the beginning itself, this altered self, it is recognized. The cells in our body tell that they are not exactly our self cells. So they see that, it is that something different from what is normal. That is when the cells are, the immune system comes into action, and that non-self or the tumor cells for that matter, or the cancer cells, beginning, they are tried to be eliminated out of the system. Of course, it is a different story altogether that, it is not exactly taking place. And, once, a lot of cell cycles take place, and, a lot of mismatch DNA repair is failing, a lot of things come into action. That is when the cell turn into more cancerous and it spreads. But initially, our body tries to fight these non-self cells. That is what is meant by elimination of altered self. And how is it again damaging us? Our immune system damages us, causes hurt to us also. How? Collateral damage is inflammation. You have a foreign particle, some nail or something got stuck inside. So you know that pus cell, there is a basic swelling in that particular area, and then it causes pus. It causes a lot of pain, a lot of discomfort. So this is the collateral damage that you're talking about. And you have damage to self, that is hypersensitivity or autoimmunity. Certain diseases, farmer's lung, rheumatoid arthritis, and you have systemic lupus erythematosus. All of these, what happens is our cells, normal basic body cells in certain places that are there, are considered as foreign, because the immune system is reading it as foreign, because of which it acts against our own self cells, because of which there is inflammation and other side effects that are there, that manifests itself as a problem, that is a disease. Again, same thing, specific, non-specific, innate and adaptive. Cellular and humoral components are activated again or triggered. Like I told you, innate is four barriers to the infection: anatomic, physiological, phagocytic, or inflammatory. You can see skin, physical barriers, that is the skin, and then the mucosal membrane, basically, the squamous epithelium, basic skin, it's protecting us. It's a barrier, it's a physical barrier. And then the mucosal membrane, so that, the mechanism is peristalsis, mucociliary elevator. And then your tears, they are one kind of barrier wherein, whatever falls into your eye, they are flushed out because of tears. So, and then mucous membrane protects the respiratory parts, gastrointestinal, urogenital parts for that, from microbes, in fact. When it comes to, just a second. Then you have the sweat, also antimicrobial. Tears and saliva have lysozyme in them. In fact, these are the physiological barriers, basically. Gastric juices, and then what do you call it? Saliva prevent this harmful pathogens that are present in the food from actually causing us some kind of harm. And then you have the cellular barriers, and you have this, what do you call it? Cytokine barriers. Cellular barriers are again inside the blood cells. So they include the polymorphonuclear phagocytes, so the neutrophils, the monocytes, natural killer cells. And the macrophages. They help in killing and eating the cells. Macrophages, kill and ingest the microbes, in fact. And then you have the cytokine immunity, basically. That is a specific, that is what, humoral and cell mediated. So cytokines come into that category. In this, what happens is whenever a virus or a bacteria enters, then few of the WBCs, They form these lymphokines, interferons, tumor necrosis factors, etc. So they're all antiviral proteins, and then they help in protecting the the body against these foreign invaders. So when it comes to this, see, as you can see, opsonin, antimicrobial, defensins, sweat, all of these come. Then that's again, acquired or specific immune system is that, this is basically the third line of defense in the body. What happens is once the primary, once a pathogen enters into the body, the primary response is there. Immediately what will happen is your innate immunity kicks in. Once this is done, what happens is secondary line of defense is the adaptive immune system. So because of which you are able to see that, you recognize the antigen. And then once you have done that, you should be able to do the third line. That is, defense. That is when your antibodies and the activation of humoral and the cell-mediated immunity kicks in. So subsequently, the same pathogen enters into the body again. Now, this time the body will not keep quiet. It will show its full intensity against this, pathogen. So the primary and the secondary immune systems or the responses are carried out with help of B lymphocytes and P lymphocytes. Recognition, yes, this is a foreign body. Then activation. The first response is going to be slow because we do not know how much of, see, for example, simple. Until and unless you know how much of dosage of a medicine you have to give, you'll not be able to know that without starting to give the medicine. It's always a trial and error. Then afterwards, then the B lymphocytes produces antibodies. Then the T cells secrete that which that don't secrete these antibodies, help the B cells to produce the antibodies. So this is all about the first line, and see, anatomical barriers, mechanical, chemical, biological, humoral, and cell-mediated ones. This is the first line of defense. I'll be sharing these PPTs with you, so you need not worry at all. Again, skin, sweat, tears, and others include the saliva, mucus, stomach acids. So this is the innate response that comes. And once the innate response is done, then the acquired response kicks in. And because of which, the effect against it is going to be more. Now, what exactly is active and passive immunity now? Now, a person gets active immunity after infection, right? So when an organism is exposed or a person or a thing or an animal is exposed to antigens, so these antibodies are produced. This first response is known as the active immunity. So after infection, that is why I said artificial active immunity is immunization. That is why involves the use of vaccines. They're available to induce long-lived active immunity in a person. Passive immunity is you're not directly producing the antibodies as such. So what happens is mother passes this kind of immunity through pregnancy. These antibodies will protect the baby for a short period of time right after the birth, because that is when only after the your immune system develops better only after exposure to pathogens around, which is not taking place. So the womb, the uterus, and the whole amniotic sac, is all protected very well from all kinds of infections. So crossing the blood barrier, the placental barrier is difficult. So when if that happens, that is when you've got to be a little extra careful. Zika virus is one wherein it causes microencephalitis. So that is where these pathogens are able to cross the placental barrier and infect the babies which are growing inside, in the pregnancy itself. We'll come to that later. So as such, what happens is this is when ready-made antibodies are directly provided to protect the body against foreign agents. Please refer your textbook. Same thing is there. Then antibodies, if they're directly available, then that is known as passive immunity. So colostrum, whatever the first milk that comes out of the mother is known as colostrum. In fact, any kind of mammal, the first milk that comes out of any mammal for that matter, be it human beings or what do you call it, your fetal that is your calves, or for that matter tigers, lions, you name it, all the mammals produce milk. So the first milk that comes out is known as colostrum. Your junnu that you get, I do not know how many of you know Telugu, but that junnu palu is nothing but colostrum that you're having. It's rich in antibodies. It's the best of the nutrition that is available for the calf, in fact, as soon as it is born. So that's what. So the yellowish fluid termed as colostrum secreted by the mother during the initial days of lactation is abundant in the antibodies, that is IgA, that serve to protect the infant. Then fetus also receives certain antibodies from the mother through the placenta during the pregnancy. So you have IgG, if I'm not wrong, which actually crosses the placental barrier. So that gives a little bit of immunity to the growing baby. And then you have, if a person is infected with certain toxins, for that matter, tetanus, so the antibodies or the antitoxin is directly injected. Snake bites, then antibodies against the snake bites are immediately injected. In fact, anti-venom as it is known as. So all this comes under the passive immunization. So occurs when prepared antibodies are given to that individual. So as you can see, same thing that I have told you. So when it comes to the differences between active and passive immunity, I think it's given in a very good, it's given in your book textbook basically. Active versus passive immunity produced by the immune system of the host active. Passive is received passively by the host, and the immune system of the host is not participating in it. Then when it comes to active immunity, it is induced by infection or by contact with the immunogen, whereas passive immunity is you're getting ready-made antibodies. So immune response in the active immunity is not short-lived and it is effective. Whereas passive immunity is only for a short amount of time, so it is not very effective as such. And when it comes to the active immunity, you have an immunological memory present. Subsequent challenge with booster doses is much more effective, in fact. Then there is no immunological memory in the passive memory, in fact, and subsequent administration of antibody is less effective because the immunes are the antigens are eliminated. Serves no purpose in immunodeficient host. Active immunity is not going to help if your immune system is low. Whereas when an immune system is low in a person, if you give passive immunity, that is, you give antibodies into that kind of a person, it is going to help him or her. And active immunity is used for prophylaxis to increase the body resistance, and passive immunity is used for treatment of acute infection. This is all about the differences between active and passive immunity. Now you have hematopoiesis. Now this is very important for the reason that your stem cells, whatever are there, they help in formation of the blood cells that are present. Your WBCs, your RBCs, all of them, your T cells, B cells, all of them come from the hematopoiesis, that is formation and development of blood cells. So the hematopoietic stem cells are present in the medulla of the bone marrow. All of them arise from one particular cell known as hematopoietic stem cell. This cell, that is why you have a lot of stem cell banks. This cell is able to produce all the rest of the cells that are present inside the body. So it is known as pluripotent or multipotent. So it is able to differentiate into any other cell that is required. So what happens is in humans, hematopoiesis begins in the embryonic yolk sac itself during the first few weeks of development. Here, the yolk sac stem cells differentiate into pre-erythroid cells. This contain the embryonic hemoglobin. And then in the third month of gestation, that is in the third month of pregnancy, this hematopoietic stem cells migrate from the yolk sac to the fetal liver and then to the spleen. Then these two organs play a very important role in the hematopoiesis, that is generation of the blood cells for the fetus. From the third to the seventh month of the gestation, that is what when these cells go into the liver and the spleen of the fetus, and then they help in growing into all the other cells. After that, differentiation of the hematopoietic stem cells in the bone marrow of the fetus becomes a major factor. Now, this goes on, it occurs in if at all there's any problem during the adult stage, then hematopoiesis can also occur in the liver. This is known as extramedullary hematopoiesis. That is why your hematopoietic stem cells are known as multipotent. That is, they can change into any other cell that you want, neutrophil, basophil for that matter, or pluripotent. So thereby they can generate granulocytes and agranulocytes. So erythrocytes, granulocytes, monocytes, mast cells, lymphocytes, megakaryocytes, all of these are the cells that are being produced by the one single hematopoietic stem cell. So there are 2 to 3 different pathways that are there. As you can see, the stem cells give rise to the myeloid progenitor cells and the lymphoid stem cells or lymphoid progenitor cells for that matter. These gives rise to the granulocytes and the lymphocytes, or agranulocytes and granulocytes, what do you call it? You can call them later, we'll come to that. So in lymphocytes, you have B cell progenitor that gives rise to B cells, plasma cells and memory cells. T cell progenitor that gives rise to T helper cells and T cytotoxic cells. TC, that is cytotoxic T cells and T helper cells. These helper cells help the other T cells. And then you have the natural killer cells. Whereas the myeloid progenitor cells give rise to the granulocytes that include neutrophils, eosinophils, basophils, and mast cells, and agranulocytes that are the monocytes. Then you have the dendritic cells and the macrophages as well. So you have a similar diagram that is present in your textbook. In fact, it's a better diagram in fact. So you can see that a common myeloid progenitor stem cell and common lymphoid progenitor cell is there. So this common myeloid progenitor cell is differentiated into a megakaryocyte. Like you can see your textbook. Megakaryocyte erythroid progenitor cell, which gives rise to the RBC and the platelets. And then granulocyte macrophage progenitor that give rise to the neutrophils, macrophages, eosinophils, basophils. Then the lymphoid ones, as I told you, T cells, B cells, and the natural killer cells. So you have different lineages over here. So then, as you can see, the progenitor commitment depends on the acquisition of the responsiveness to the particular growth factors and cytokines. You have a lot of growth factors that are involved which give rise to these cells. Then the immature precursor cells themselves then develop into the progenitor cells. Then after maturation, then they give rise to the matured and the completely developed cells. In the bone marrow, hematopoietic stem cells grow mature when they on the presence of the stromal cells, which are non-hematopoietic cells, but they help in the growth and the differentiation of these hematopoietic cells. So the stromal cells include the fat, the endothelial cells, the fibroblasts, and the macrophages. Now, these also help in the differentiation of hematopoietic stem cells by providing this hematopoietic inducing microenvironment. We are talking inside the bone marrow, the stromal cells provide this kind of environment so that the hematopoietic stem cells are able to differentiate. So then you have the colony stimulating factors or CSFs that come into place. These are named because they have the ability to induce the formation of the different cell lines. When I talk about T cell and its line, So I talk about the T cell progenitor line. So the cell line should be able to give T helper cells and cytotoxic T cells. So this colony T cell, colony stimulating factor should be there, so that this gives rise to the progenitor cells and the cell line subsequently. So another important hematopoietic cytokines are the glycoproteins erythropoietin, which are produced by the kidney. This erythropoietin helps in the development, final or terminal development of all the erythrocytes, regulates the production of the RBCs, and then you have the colony stimulating factors that actually go bind to the receptors of the stem cells. So stem cells also have receptors which are able to help in binding to the colony stimulating factors. So you have different kind of colony stimulating factors which are multi-lineage CSF. You have interleukins, you have granulocytes and macrophages, and then you have granulocyte CSF, GCSF, that is GCSF as it is known as, or MCSF, that is your macrophage colony stimulating factor. So these are the different kinds of like colony stimulating factors that are present. Now, when it comes to the cells in the immune system, you get to see that they are coming from bone marrow. We have already learned about it. Now, the cells of the immune system, sorry about that. So all the cells originate from the bone marrow. Now, these cells make up either the innate system or the adaptive system. Now, these include the phagocytic cells, that is, they engulf and kill the foreign body and then make them into small pieces, so that they are then that waste is taken out, flushed out of the cell, and this comes out through the lymphatic ducts and all that. So these include the monocytes and the macrophages, and polymorphonuclear phagocytes. Then they include the natural killer cells, that is NK cells, basophils, mast cells, eosinophils, and the platelets. Now they include this phagocytic cells, and cells that make up the adaptive or the specific immune system include the B cells and the T cells. So humoral immunity is what we are talking about over here. So B cells differentiate into, again, memory cells and the plasma cells. Now you have a very specialized subset which is known as APCs, that is antigen presenting cells. Now they play a very important role in innate immunity, and they help by they go activate the helper T cells, and then once they do that, these cells are the dendritic cells and the macrophages basically. So this will help, these APCs are expressed on the cell surface molecule encoded by genes in the major histocompatibility complex, which is actually referred to as the MHC complex. So you have class 1 and class 2 MHC complex. We'll go to that later. So B lymphocytes also express the class 2 MHC molecules, which function as the APCs. And so, but then again, it is not considered as innate. It is an acquired immune system for that matter. In addition, you have thymus cells, that is the thymic epithelial cells, and they also are able to express this class 2 major histocompatibility molecules, and they function as the APCs. Now, this is about your cells that are there. So we'll see about, again, that's what monocytic T cells, helper T cells. So you have granulocytic and monocytic immune system, myeloid, lymphoid. Myeloid has granulocytes and monocytes. So neutrophils, basophils, and eosinophils come under the granulocytes. Monocytes include the macrophages, Kupffer cells, dendritic cells, even the mast cells, and even the welcome. Then when it comes to the lymphoid cells, you have the T cells, B cells, and natural killer cells. T cells help the cells suppress. Then you have the cytotoxic T cells. B cells are differentiated into memory and the plasma cells. Memory cells is only to remember. Its function is to remember that particular antigen for lifetime. So when it comes to the lymphoid cells, you get to see that they include, they are this lymphocytes are WBCs basically, having only with one fundamental importance in the immune system, that is to determine the specificity of that particular immune response to the infection that is present. So it's about 20% of the body's leukocytes, that is WBCs. 99% of it is present in the lymph nodes. In fact, that is why they're known as the lymphoid cells. So we have your the lymphatic system in the body. You have the blood vascular system in the body, and you have the skeletal system in the body, and you have the different other, of course, I need not talk about the different organs and their particular systems. But basically, if you see, you have a lymphatic system, and then you have the blood vascular system along along with the nervous system and the skeletal system. So 99% of the lymphatic system has these lymph cells, and the lymph nodes for that matter. And if it is not present in the active form, it is known as the naive cells or the null cells. And the nucleus occupies almost the entire cell for that matter. So these include the, as you can see again, what do you call it, cytotoxic T cells, helper T cells, and B cells. So they are found in circulation, present in the lymphoid organs and tissues such as spleen, tonsils, lymph nodes, which are initial responses that is likely to occur. Two types are there: B cells, B lymphocytes and T lymphocytes. Both of them come from the stem cells in the bone marrow, initially similar in appearance. And some lymphocytes migrate to the thymus gland, mature into the T cells. Others remain in the bone marrow, developed into the B cells. So this is how they come. Again, hematopoietic stem cells, then multipotent progenitor cells, common lymphoid precursor cells, pre-cells, pre-B cells, immature B cells, and this is how B lymphocytes come. This is activated follicular B cell, activated B cell to memory and plasma cells. So first stages of B cell development takes place in the bone marrow, and it is known as B because first time it was discovered in the bursa of Fabricius, which is in birds basically, which is an organ, something similar to the thymus in our body. So that's how. And for all the B cell development, interleukins are very much necessary and important for that matter. So you have the early lymphoid progenitor cells. Then from that common lymphoid progenitor cells, that give rise to the natural killer cells, T cells, and the macrophages. And then the myeloid progenitor cell gives rise to other parts, that is the neutrophils, monophagocytes, erythrocytes, eosinophils, etc. And common lymphoid progenitor 2 is responsible for the B cell lineage. So what happens is the first immature B cells are produced. Then you have different kinds of signaling pathways such as the Notch, which is necessary, Notch N1 signaling pathway necessary for the thymus cell or the T cell development. After interaction, you have antigens after interactions with the antigens, that is the B lymphocytes then get differentiated into the plasma cells and the memory cells, and the B lymphocytes also serve as antigen presenting cells. So this is all about the B cells basically. So you have if the BCR complex is there, so which is a molecular complex built in by immunoglobulins M and D. Then you have two additional immunoglobulin accessories such as Ig alpha and Ig beta for that matter. And CD40, which is a transmembrane glycoprotein type 1 receptor. These are all different receptors that are present, and then TNF factor receptor, tumor necrosis factor receptor superfamily. So all these are expressed on a great variety of cells, that is, monocytes, antigen presenting cells, endothelial cells, and they're all present during the stages of all the stages of the B cells basically. Now, like I told you, let me see what is the next one. So you have granulocytes and agranulocytes definitions basically. I'll come back to this in some time. So when it comes to the T cells, you get to see that the T cells can either get differentiated into the cytotoxic T cells or the helper T cells. They are like, then helper T cells are of two types, that is H1 and H2, and they are identified by the presence of a CD2 molecule on the T cell surface. So when CD2 molecule is present on the T cell, then it is known as the helper T cell. So either it can be H1 type or H2 type. And another group of membrane-anchored molecules were identified that comprises of the polypeptide CD3. So when this protein is present on the T helper cells, then these molecules associate on the cell surface with the T cell antigen receptor. It's a group of molecules to be used by the T cells to recognize, interact with an antigen in the form of amino acids. Proteins are basically blocks of amino acids put together. So any antigen receptor you're talking about, or anything for that matter, all of them are proteinaceous in nature. Of course, you have glycogen and fats associated with it. It can be lipoprotein, but basically it's a protein. So if that is present along with the T cell antigen receptor, then the antigen is bound to the T cell, with which the next subsequent reactions in the immune system occur. Some T cells express CD4 molecule. So we have seen CD2, CD3 and CD4 right now, along with CD8 molecules. So you have CD4 molecules and CD8 molecules on the cells. When they are there, then CD4 plus T cells are known as helper cells because they help the B cells to produce the antibodies. And CD8 plus T cells, they are known as cytotoxic T cells because they are capable of killing by themselves. They are capable of killing the antigen by themselves. That's what I mean. Most mature T cells express the membrane molecules such as CD28, another CD that is molecule that is present. It is a receptor for stimulating the B7 molecules that are actually present on the B cells and other antigen presenting cells such as the CD45, which is a signal transduction molecule. So CD here stands for cluster of differentiation. So cluster of differentiation means how do you differentiate between all the T cells is because of these particular proteins and molecules that are present. They are named as CD2, CD3, CD8, CD4, and then you have CD28 and CD45, which are all proteins basically. Now, when we come to, then we have the natural killer cells. Let me come to that. I'll have to present what system, T system. So they are known as null cells or natural killer cells. So these cells can actually kill some tumor cells. There are about 5 to 10% of the lymphocytes that are present in the human peripheral blood basically, and they have only a limited range to kill the target tumor cells. So these molecules are products of MH1, that is major histocompatibility complex, so 1 for that matter. And the NK cells, they're also known as NK cells predominantly. They destroy the target cells through activation of molecules, and these molecules are the perforins and granzymes. So NK cells express CD16 membrane receptor for the carboxy terminal end of the Ig immunoglobulin molecules, which is actually known as the Fc region. Then they can attach themselves to the antibodies, subsequently destroy the targeted cells. So NK cells play an important role against the tumor cells basically, and this is known as the antibody dependent cell mediated cytotoxicity. That is, you have an antibody involved, that is immunoglobulin G, wherein this NK cells go and attach themselves to the carboxy terminal end of the immunoglobulin, that is the Fc region. So you have an antibody involved, and then you have the T cells also involved in this. The natural killer cells. So that is known as the antibody-dependent cell-mediated cytotoxicity. They play an important role against the tumors. And example is that, in your textbook, they have given the Shediac-Higashi syndrome, which is an autosomal recessive disorder. That is, only both the parents are autosomal and they have the same gene carrying for this particular disorder. It should be recessive and it should be dominant in both the genes. That is when, what I mean is, both the parents should carry the gene for the progeny to express this particular disorder. That is what is known as autosomal recessive disorder, wherein both the parents should have this gene together. And this is associated with impairment in neutrophils, macrophages, and NK cells, because of which there is an increased incidence of lymphomas, which is a cancer or a tumor in the lymphatic system. So when it comes to the monocytes, that is mononuclear cells, they are known as monocytes in the bloodstream, and they are also known as M5 cells in the tissues. And then the monocytes are actually 5 to 10 times smaller than that of the M5 tissue cells in the tissues, basically. So they get differentiated into either macrophages or into dendritic cells. They're completely present all over the body, and then in certain tissues what they do is they are present as such, so they are known they become fixed macrophages, whereas in others they are motile in nature and they are free, so they are known as macrophages when they travel all over the body, for that matter. They go by amoeboid movement, and macrophage-like cells, they help in for serving different functions in the different tissues. So you have alveolar macrophages in the lungs, Kupffer cells in the liver, histocytes in the connective tissues, mesangial cells in the kidney, microglial cells in the brain, osteoclasts in the bone. So then how do they get activated? They also help in the presentation of the antigen through the MHC class II molecules. So they get activated with interferon gamma, which is secreted by activated T helper cells. So macrophages also are see, this is the basic difference between monocyte and macrophage. All the most of it is taken by the nucleus occupies a lot of place for that matter. And you have the lysozyme enzyme, you have a monocyte with the nucleus which is bean shaped, you have the lysozyme and the phagosome. Over here it is, the nucleus is round in shape, you have a phagosome over here, you have amoeboid movement, so because of which it has pseudopodia. This is a phagosome, lysosomes, phagolysosomes. So this is how basic generation, difference between monocytes and macrophages. So they are effective antigen presenting cells. So basically they have with the help of class II MHC molecules, they help in presenting this antigen. This is the bacteria through phagosome, that is and through lysosome it comes. And this is the exocyte. Then you have MHC class II molecules. So this antigenic peptide is being presented over here, and this is the exocytose degraded material. So this is about the monocytes for that matter. So when it comes to that, like I told you, activated macrophages also synthesize lysosomes and other hydrolytic enzymes. Then they do not require oxygen. So in addition, they produce a group of antimicrobial or cytotoxic peptides known as defensins. These molecules are cysteine rich. The amino acid cysteine is present, which is a cationic peptide, cationic amino acid for that matter, has around 29 to 35 amino acid residues. Defensins can kill a variety of bacteria, including the Strep aureus, Staph aureus, Streptococcus pneumoniae, E. coli, etc. So activated macrophages also secrete. You have TNF alpha, which are cytokines basically, TNF alpha, beta, gamma. There are 3 different kinds of tumor necrosis factors. Anyway, TNF alpha is secreted by this monocytes. Now you have granulocytic cells. They include the neutrophils, the what do you call it? I'll come to that. They include the neutrophils, eosinophils, and basophils. Right. So granulocytes, they have the cells are granular in nature. They have small granules which can be basically dyed. So like either using acidic dyes or basic dyes. So when they stain, you can see that there are basic granules because of which they are known as granulocytes. So the granulocytes are classified as neutrophils, eosinophils, basophils, and neutrophils and basophils are phagocytic in nature. Neutrophils and eosinophils have phagocytic in nature, whereas basophils are not phagocytic. So neutrophils constitute the largest number of WBCs that are present, that is around 50 to 70%, and of the circulating WBCs basically. And then numerous cells such as the eosinophils are around 1 to 3%, basophils are less than 1%. So if you actually see your any kind of complete blood picture CBP report, you get to see these, they are given. So basophils 0 to 1. So this is what you see. Out of 100 cells, how many basophils are there? Only 0 to 1. Eosinophils maybe around 1 to 3 if it is present, that is normal. Whereas neutrophils should be around 50 to 70% of the circulating WBCs. So neutrophils are again multi-lobed, and then I will be finishing till the dendritic cells and will be stopping. So that is another 5 minutes. Next class, I will do the organs of immune system, and then I will also do the antigens. It is a small topic, the antigens and the antibodies for that matter. So antigens and the organs of the immune system will be dealt with in the next class. So right now, let me finish with the granulocytes and up to the dendritic cells. So please give me 5 minutes. All right. So when you come to see the neutrophils, they have multi-lobed nucleus, as you can see, horseshoe shaped or whatever it is, they are multi-lobed nucleus, and it gets stained with both acidic and basic dyes. Often they are also known as polymorphonuclear leukocytes, PMN, because nucleus. So the nucleus has many lobes. Because of the presence of the multi-lobed nucleus, it's known as polymorphonuclear leukocytes. They are the most common WBCs present, and the primary function is to remove a microorganism from the blood, known as this, and this process is known as phagocytosis. They help in providing protection and from a variety of microorganisms for that matter. Most important fighters for that in the blood basically. They're quite large cells, 10 to 20 millimeters in diameter, but they live only for 2 to 3 days. So whenever there's a bacterial or a viral infection, the neutrophil count will increase. And neutrophils are present all over in the blood, and they quickly move to the site of infection or injury to fight the pathogens off. So whenever there is a local injury or tissue trauma, infection, whatever it is, mosquito bite for that matter, then the neutrophils become activated. They come from the capillaries and the post-capillary venules into the tissues, and then by a response through chemotoxins, chemokines, the neutrophils act. The next is eosinophils. Are you able to see this picture? Let me enlarge it. You have quite a number of pathways that actually activate the eosinophils. So eosinophils are bilobed. They have a granulated cytoplasm which stains with an acidic dye which is known as eosin red, because of which these are known as eosinophils. Eosinophils are motile phagocytic cells. They migrate from the blood into the tissue spaces. Then in the tissue spaces, they play a role in the defense against the parasitic organisms, especially which are present in the tissue spaces. Then the secreted contents of these eosinophilic granules damages the parasitic membrane. So primary granules that are present, they contain the Charcot-Leyden crystal protein, and then you have the secondary granules. They have a crystalline core of major basic protein. So these granules have major basic protein, and you have the Charcot-Leyden crystal protein basically. These cause the damage to the parasites. So especially during parasitic infection, you can see the count of eosinophils that are increased. And you also have matrix eosinophil cationic protein, eosinophil derived neurotoxin EDN, and eosinophil peroxidase. So this major basic protein, whatever is present, is highly toxic to the parasites. So there are different kinds of pathways that actually help in triggering or activating the eosinophils. They include the growth factors, interleukins, then colony stimulating factors. Then you have so others as well. So all these stimulate or activate the eosinophils, because of which it gives a defense against the mucosal pathogens by releasing all the proteins, or it is pro-inflammatory, that is tissue damage and remodeling, because of which mast cells are activated and then it survives. And you also have immunoregulatory function of the eosinophils, because of which T cells, B cells, and dendritic cells are also being activated. Now, when it comes to the basophils and the mast cells, you get to see that both mast cells and basophils are from the hematopoietic stem cells in the bone marrow. Their progenitor cells are they come through with the influence of interleukin 3, IL-3. Now basophils get differentiated in the bone marrow, and then so this is because of interleukin 3 and cytokine transforming growth factor beta. So these two help in the differentiation of the B cells in the bone marrow. Then they enter the circulation, that is the basophils enter into the blood circulation. They are less than 0.2% of the WBCs. And then you have mast cells that are present. These mast cells get differentiated because of the stem cell factors, SCF. Then this is all taking place in the bone marrow. Once they leave the bone marrow as precursors, they go into the tissues, they proliferate and differentiate into the mature mast cells. So whatever the SCF that is present, stem cell factors, is producing immature mast cells, which leaves or migrates into the tissues, wherein they differentiate into the mature mast cells. So basophil again has lobed nucleus. Do I have basophils over here right now? No. Unfortunately, I do not have basophils. So let us just please listen or look into your textbooks. The basophils has a lobed nucleus, heavily granulated cytoplasm, baso. So because of which it is because of basic dyes, that is methylene blue, it can stain the basophils. So that is why it is known as basophil as such, because basic dye can stain it. So they are non-phagocytic granulocytes. This is how a basophil looks. So they are non-granulocytic phagocytes, non-phagocytic granulocytes, and that function by releasing active substances from the cytoplasmic granules. This plays a major role in allergy or allergic responses. You also have mast cells that are found in skin, connective tissues of various organs, mucosal epithelial tissue of the respiratory, urogenitary tract, or digestive tract. So this, along with the basophils, help in allergies, defense against allergies. Now, basophil types are they again play a role in the allergic reactions, inflammatory or inflammation, responses against cancer or parasites, then angiogenesis, that is formation of new blood vessels and tissue remodeling. They are the only cells on the membranes that have a high affinity receptor for immunoglobulin E. So IgE is associated with basophils because of the receptor, high affinity receptor that is present. So this is about the basophils. Now let us look into the dendritic cells. Now they are irregular shaped, and the dendritic cells for that matter, they are in irregular shaped. And then you get to see that they are of different kinds. They are they have thread-like finger-like cytoplasm, and this surrounds the cell membrane. And then they are found in most of the tissues in the body, and they are helping in. They are also the professional antigen-presenting cells, or the APCs. Different types, like I told you, Langerhans cells found in the skin, interstitial dendritic cells that are present in heart, lungs, liver, intestines. Then you have the myeloid dendritic cells or the myeloid cells, and the lymphoid. So that is MDC1 and MDC2, and then you have the lymphoid dendritic cells. Each arises from the dendritic stem, stem cell precursor for that matter, and uh, different pathways give rise to in different location gives rise to different kinds of dendritic cells. So whenever there is an invasion because of microbes, or because there is inflammation, or immature forms of Langerhans cells and interstitial dendritic cells, they go into that place into draining the lymph nodes. Then they make this presentation. They are antigen presenting cells. So they present the antigens to the T helper cells. So this triggers the immune system pathways, either the humoral immunity or the cell mediated immunity. So myeloid dendritic cells have high levels of receptors in the cell membranes. So this gives rise to the innate immunity, important in binding the toxic products of the bacteria, so that they bind, present, so that once that is then the humoral immunity, that is antibodies, come into place, or the cell mediated immunity, so that the different kind of cells, natural killer cells, the eosinophils, the neutrophils come into place, and then they neutralize that particular pathogen or antigen. So this is all about your cells of the immune system for that matter. I will end my class right now. So tomorrow you're going to see the organs of immune system. Then you're going to see the antigens as well. So this is all for today. I'm going to stop sharing. And right now, whatever you have seen, I will just be sharing these PowerPoints for you later. So hopefully by tomorrow, I'll be able to give you your complete this particular lesson and antigens, PowerPoints. So whatever class I'm taking, that day's PowerPoints will be presented to you either that day itself or the next day. All right. So I'm going to end the class right now. The class is over. You have your class from 1:30 to 2:30, right? Okay, then you can all leave for your next class. Thank you.